Hello and welcome back. Thanks for coming back. We were talking about the innate immune system and how they coordinate and how they communicate. And so we're going to jump right back in. We were saying that there's three different types of cell communication: cytokines, surface receptors, and cell adhesion molecules. So that's where we were. Hope you're doing all right today. All right, so this very first one, let's talk about cytokines. And cyto means cell, kine means to move. So these are the voices of the cell. And so there's always going to be a sender. So there's a cell that is producing a cytokine, and it's going to be in response to something, not just goes, hey, it's Thursday, let's do this. Like I said, they don't say, what's up? So there's always a talker, and then there's always going to be a receiver, so a listener. And so they're going to have a receptor for this particular cytokine. And so they've given them different names depending upon what they do. So what kind of action is that cell going to take? What is the effect? So chemokines, and so that's telling you it's a chemical. Kine means to move. So these are chemicals that are going to make cells move. And so these are signals for chemotaxis. And usually it's going to be positive chemotaxis. So it's going to call immune cells to the area. The next one we mentioned when we were talking about hematopoiesis and the production of leukocytes, white blood cells, they are going to be stimulated to be made and to differentiate by colony stimulating factors. So that's just another example of a cytokine. Interferon, there's different interferons. We're going to talk about a special one that is released in response to a viral infection. And so it's involved in viral infections, and then others are also involved in regulating immune responses. Interleukins, and it's kind of telling you in between white blood cells. So these are produced by leukocytes, and they play an important role both in the innate and the adaptive immune system. This other one has a very cool name, tumor necrosis factor. And so it got its name. Necrosis means death, so death of tumor cells. This particular cytokine is capable of all kinds of things. It is a mediator of inflammation. It can bring about apoptosis or programmed cell death. Usually, groups of cytokines are going to work either in a sequence or they're going to act together to generate a response. And just a beautiful example, and this is kind of what's happening. It happens in the case of COVID-19. These three cytokines work together for inflammation, so that big immune response that is occurring in COVID-19 that's causing complications. So interleukin 1, interleukin 6, and tumor necrosis factor are pro-inflammatory cytokines. So this next section, we are talking about surface receptors. And so types of surface receptors, they've given this fun little name of PRRs, so pattern recognition receptors. And these are going to be on our white blood cells of the innate immune system, and this is how they recognize something that's foreign, so something that is not us. And when they recognize these things, that's going to lead them to cytokine secretion, and that's going to make something happen. And in particular, it's going to cause inflammation. So think about it, pattern recognition receptors, so those are the eyes. Your book says ears. I'm just going to go with eyes and a magnifying glass. And if I can get my animation to work, there we go. One type of pattern recognized by these types of receptors are called PAMPs, so pathogen-associated molecular patterns. Your book also says sometimes they're called microbial-associated molecular patterns, but I'm just going to use one. We're trying to eliminate pathogens from the body, so we're just going to go with PAMPs. So pathogen-associated molecular patterns. So that's one type of pattern that they can recognize. Another thing that they can recognize is just cellular damage. And so those are called DAMPs. So damage-associated molecular patterns. And so when these pattern recognition receptors recognize these things, and we're going to look at different ones in just a minute, what they're going to do is they're going to respond by releasing cytokines. And I told you that often cytokines are going to act in sequence and in tandem together. And I gave you this example of interleukin 1, interleukin 6, and tumor necrosis factor. We know that all of those lead to inflammation. And we're not going to talk about inflammation in this particular lecture, but I will get to it next lecture. Okay, so we are going to watch this video, and so this video is going to show you a group or a type of pattern recognition receptors called toll-like receptors. And so just kind of sit back, just kind of watch it, and then we're going to unpack it after this video.
Cells of the immune system, such as macrophages and dendritic cells, are the first line of defense in recognizing pathogens of various kinds. These cells have developed several kinds of receptors for recognizing different kinds of pathogen-associated molecular patterns, known as PAMPs. There are different classes of these proteins that recognize different types of PAMPs. Toll-like receptors, or TLRs, are composed of multiple leucine-rich repeats that are useful for recognizing various PAMPs. TLRs are membrane-associated proteins. Some are located on the surface of the cell, while others are located on endocytic vesicles, where they survey the degraded contents of pathogens taken up by endocytosis. Each member of the TLR family recognizes different kinds of PAMPs. For example, TLR5 recognizes flagellin, which is a highly conserved constituent of the bacterial flagellum. Bacterial genomes contain methylated CpG oligonucleotide motifs, which are recognized by TLR9 once the genome has been degraded in the lysosome. TLR6 and TLR2 are a dimer that recognize diacyl lipopeptides. TLR1 and TLR2 are a dimer that recognize triacyl lipopeptides, and TLR4 recognizes lipopolysaccharide, LPS, a component of gram-negative bacteria. Like TLR9, TLR3 and TLR7 are located on endocytic vesicles and recognize double-stranded RNA and single-stranded RNA, respectively. When any TLR is activated, it sends a signal to the nucleus by activating transcription factors. Not all pathogens, however, live in the extracellular space or are phagocytosed. Some pathogens, such as viruses, exist and replicate in the cytosol. There are at least two classes of receptors that can detect pathogens in the cytosol and signal their presence to the immune system. One class of such receptors are members of the nucleotide oligomerization domain family, or NOD proteins. For example, the NOD2 protein, which is located in the cytosol, can detect bacterial proteoglycans of intracellular bacteria. When the NOD2 protein recognizes its ligand, the muramyl dipeptide, it sends a signal to the nucleus to activate transcription. Finally, there is a class of intracellular receptor proteins that contain an RNA helicase domain and two caspase recruitment domains, or CARD domains. One member of this family, RIG-I, recognizes double-stranded RNAs that are a component in the life cycle of many RNA viruses. This class of proteins also sends a signal to the nucleus, but unlike TLRs and NODs, it activates the production of type 1 interferons. In all, the toll-like receptors, NOD proteins, and the RNA helicase CARD domain family provide the innate immune system with the ability to detect both extracellular and intracellular pathogens and to activate an immune response against them.
Okay, so let's break this down. First, we'll start off with where are the pattern recognition receptors? And we just watched the video, but let's go ahead. So pattern recognition receptors, if they're toll-like receptors, they are going to be on the surface of the cell membrane. And so they're going to detect microbial components present in the cell surroundings. There are also some pattern recognition receptors in the phagosomes and endosomes. So when something is brought into a phagocytic cell, so a macrophage, a dendritic cell, a neutrophil, once they are inside that phagosome, they can fuse with a lysosome, and anything that's in that vesicle is going to get degraded, and then that's where you're going to get binding to those pattern recognition receptors. And then there's also other classes of pattern recognition receptors that are completely intracellular, like RIGs and NODs. We'll look at them in a second. So they would detect cell damage as well as microbial components in the cell cytoplasm, so viruses or bacteria that are capable of living in the cytoplasm. What they can see, or what the patterns that they recognize, are things that we don't have, so things that aren't self. So peptidoglycan, which is going to be in all bacteria, lipoteichoic acids, which are part of gram-positive walls, LPS, which is part of the gram-negative bacterium, lipoproteins. There's different ones, but some of them are specifics to Staphylococcus aureus. Flagellin is part of flagella. Bacterial DNA is different from our DNA, it has different methylation and different sequences that we don't have that can be recognized by these pattern recognition receptors. Double-stranded RNA is something we don't have, so that's going to be indicative of a viral infection, and when that's recognized, we're going to get interferon. Single-stranded RNA, we have single-stranded RNA, but that's with that 5' methylene cap. That's different. Bacteria don't have that. Viruses won't have that. So that would be something that the cells could see. Cell damage. When a cell is damaged, they release certain chemicals, and so that too can be detected by these white blood cells of the innate immune system. So take-home message, pattern recognition receptors see and respond to things that aren't self, and they're also going to respond to cell damage. So we got the where they are, we've got the what they can see. Okay, now look at the who. So who? Tolls, rigs, and nods. That makes it easier for you. So toll-like receptors, which I never knew how they got their name until I read your textbook. Just toll meant awesome in German, and I always wondered why. I always thought it was like, oh, you have to pay a toll and you stick a coin in the slot, and that's sort of what made sense to me. But toll-like receptors are different from rigs and nods, so rig-like receptors and nod-like receptors, in the fact that they are part of a membrane. So they're going to span a membrane. They can be on the cytoplasmic membrane facing outward, so detect things in the environment. And we kind of already talked about it, so there can be specific ones for LPS, specific ones for lipoprotein, specific ones for flagellin, or they're going to be in the membrane of a vesicle. And so inside of an endosome. And so this one could detect double-stranded RNA, which you would see in viruses. This one can detect bacterial DNA because it's different from ours. This one can detect single-stranded RNA that's different from ours. And so toll-like receptors, they're anchored in a membrane. You're going to find them in those sentinel cells, those ones that are always on watch. So macrophages, dendritic cells, also have them in neutrophils. And then the other two types of receptors, these are different. There we go. So rig-like receptors and nod-like receptors, what they have in common is they are going to be in the cytoplasm. And so that's what this slide is showing you. And so it's showing you rig-like receptors detecting single-stranded RNA and rig-like receptors detecting uncapped single-stranded RNA. And so they're going to be detecting that viral RNA. And when they detect that, then we're going to get a release of interferon. And so these can be in other types of cells besides the white blood cells. They're going to be present in most cells. And so that's how a cell is going to signal to its neighbors like, hey, there's a virus around, I'm infected. Other types of receptors, so nod-like receptors, they're also in the cytoplasm. They are going to detect microbial components or cell damage. And so these are going to become really important in dendritic and macrophage cells. There's a bunch of different types. Your book kind of focused in on what happens in dendritic or macrophage cells when they detect things inside of them. So when they detect flagellin or bacterial secretions or peptidoglycan inside of them, they're going to go through a special kind of death and basically sound the alarm. So they're going to trigger inflammation and cell death, and so pyroptosis, and that is going to be in the next lecture. So I'm just kind of throwing that out there. So let's look at that interferon response. And so when the pattern recognition receptors, they detect viral RNA, that's going to cause a cell to produce interferon. So I think we're up here. So here we have a virus, we've got single-stranded RNA, double-stranded RNA, so that has been detected. And then just like on that YouTube, then we're going to get transcription factors and we're going to get production of interferon. So interferon is a type of cytokine. It's going to diffuse to neighboring cells. The effect it has on neighboring cells is going to cause those neighboring cells to make an inactive antiviral protein. And you can think of this like an inactive suicide protein. And so it's like, okay, this protein is going to remain inactive unless this neighbor cell gets infected with a virus. So kind of pop up over here. Oh, look, hey, viruses again. We have a productive infection. So either these virus particles, so these virions, they're either going to be leaving by exocytosis, they're going to be leaving by budding, they're going to be leaving by lysis of the cell. But we have a productive infection here. And so what happens when one cell releases virus particles, it can go to infect a neighboring cell. So once we have a viral infection, so we have entry into the cell, that is going to activate that antiviral protein. And so then we are going to get what's going to happen when it's activated. We're going to degrade the messenger RNA. We're going to stop protein synthesis. That infected cell will then go through programmed cell death, which is called apoptosis. And that's going to prevent the spread of that virus to the next cell. So this is one of the methods that the innate immune system uses to combat viral infection. Okay, so our last topic in this section, we're going to talk about the complement system, or sometimes it's just called complement. And it got its name because it's going to complement activities of both the adaptive and the innate immune response. And so what complement is about 25 circulating proteins, and they're made by the liver, and they circulate in the blood and tissue fluid in an inactive form. Unfortunately, kind of like the blood clotting cascade, if you can remember that from AP2, they were named in the order they were discovered, not in the order that they function. But this is going to be a chemical cascade or pathway. And so these complement proteins, they're going to split. So they have an inactive form, and then they split into fragments. And when they split into fragments, usually then those fragments will go on to split other members of the complement system. And so we're going to watch a YouTube in just a minute. There's 3 different pathways that are going to lead to the formation of sort of the common element that's going to stimulate and kick off this cascade, and that is the formation of C3 convertase. And so when we get C3 convertase, it's kind of telling you it's going to convert C3, which is in inactive form. It's going to convert C3 and split it into C3A and C3B. And then we're going to get that complement cascade, which we're going to look at in just a moment. Something I want to show you about C3 is that even in the absence of something triggering the complement cascade, C3 is somewhat unstable. And so it automatically splits into C3A and C3B. And the idea is that C3B is going to bind to microbial invaders. So just kind of tuck that away in your mind for just a couple minutes. Let's just take a look. There are 3 pathways to activate this awesome complement cascade. And one of them is that alternative pathway, which I was just mentioning. C3b is an active protein, and it can just bind to foreign cell surfaces. And I'll show you how it knows in a couple of slides. So it just binds to foreign cell surfaces. And once it binds, then we get formation of C3 convertase. I know, kind of weird. And then we're going to get our complement cascade. Another way, the second way, is the lectin pathway. And what's going to trigger this pathway, once again, this is going to be just kind of pattern recognition. There's a sugar mannose binding lectins or MBLs that they are going to bind to mannose. And mannose is in microbial cells, usually bacteria or fungi. We can make mannose too, but this is going to be on the surface of both of those classes of foreign cells. And so it's going to bind to, and then once we get binding of those mannose binding lectin proteins, then once again we go to our commonality, the formation of C3 convertase. The classical pathway, and I'm going to bring this one up again when we talk about antibodies, but antibodies are part of that adaptive immune system. They're made by activated B cells, which are plasma cells. But antibodies are incredibly specific, and so they will bind to their target. Antibodies themselves really don't have any power other than that they bind things, they cover up binding sites so they neutralize them, and they target them for destruction. And one of the ways they target them for destruction is by activating complement. And so we've got the alternative pathway, the lectin pathway, and the classical pathway. And so I like this picture. It's from the A&P book instead of yours. Yours had just tiny little writing. So what's going to happen, because we want to talk about the outcomes of activation. And so we have the C3, it splits into C3b, C3a, because we got that C3 convertase. So the three things the complement does. One is C3b binds and coats pathogens. When it does that, it enhances phagocytosis, so that cell eating. And so your book, I used to always talk about like ketchup, putting ketchup on something makes it tasty. Your book says when something is covered in C3b, it's like half a Velcro. And then there's receptors on the surface of macrophages, dendritic cells, neutrophils, eosinophils, any phagocytic cell. And so they're going to bind to the C3b, and that just gives them a handle so they can better eat them. So phagocytosis. So it enhances phagocytosis. Pop over here, and I realize we haven't talked about inflammation yet, but what it's going to do, it's going to stimulate inflammation by a release of histamine. It's going to increase blood vessel permeability, so we're going to get more blood and more white blood cells to the area. And it is going to attract phagocytes by chemotaxis. And so it's going to call those white blood cells to that area of invasion or by trauma. So inflammation kind of responds that way. And we'll hit that next lecture. The other special thing that they can do, so C5b through C9, these proteins can form a membrane attack complex and Big Mac attack. And what they can actually do is they'll actually self-assemble and put a hole in the membrane of its target. And when it does that, that's going to interrupt the integrity of that, and so the cell's going to lose fluid. It's going to lead to cell lysis is the big thing. This is more effective in fighting gram-negative bacteria because remember, gram-positives have that very thick peptidoglycan cell wall. But we have opsonization, inflammation, and cell lysis are the three things that complement does. Opsonization, inflammation, hopefully I just said all of that. Okay, so let's watch it. Don't get too caught up in the chemical cascade.
Complement consists of a group of serum proteins that activates inflammation, destroys cells, and participates in opsonization. Complement can be activated by a number of different foreign molecules. The complement proteins respond in a sequential manner, producing a cascade of reactions. The major components are C1 through C9, named in the order that they were discovered, not in the order in which they function. The complement cascade can be activated by the classical pathway or by the alternative pathway. In the classical pathway, C1 becomes activated when it binds to an antigen-antibody complex. The activated C1 then cleaves C2 into C2A and C2B, and C4 into C4A and C4B. C2B and C4B combine to form a protease called C3 convertase. C3 convertase then cleaves C3 into C3A and C3B. In the alternative pathway, antigens such as endotoxin, polysaccharides, or cell wall components react with C3b. Small amounts of C3A and C3b are constantly being formed from C3, but without activation they are soon destroyed. C3b reacts with the proteins factor B, factor D, and properdin to form a complex called C3 convertase, which cleaves C3 into C3A and C3b. Both of these pathways of complement activation follow the same sequence after cleavage of C3. C3A is involved in stimulating inflammation. C3b reacts with other complement components to form C3 convertase, which forms more C3A and C3b. C3b also attaches to the surfaces of microorganisms. Phagocytes have a binding site for C3b. Therefore, microorganisms with C3b bound to their surfaces are more susceptible to phagocytosis. Coating of bacteria to make them more susceptible to phagocytosis is called opsonization. Addition of properdin to C3 convertase results in formation of C5 convertase, which cleaves C5 into C5a and C5b. C5a enhances inflammation and acts as a chemoattractant for phagocytes. C5b reacts with other complement components including C6, C7, C8, and C9 to form a membrane attack complex. This structure forms a hole in the cell membrane and causes cells to lyse.
Sorry that popped in over the picture, but hopefully you got what you needed out of that. Okay, so we have these proteins and they're circulating in inactive form, but I told you that C3 is kind of unstable. And so there has to be some sort of regulation that prevents host cells from activating the complement system, because we wouldn't want that sort of opsonization, inflammation, or any sort of membrane attack complex. And so our host cells have a regulatory protein, so the complement regulatory protein. And so if there is C3b, it's going to attach to the host cell surface, and then the regulatory protein is going to inactivate it. And so complement won't be activated. If there is microbial cell surface, they don't have this host cell, so they don't have the complement regulatory protein, and therefore C3b will bind, and then we will get complement activation. So that's just a little safeguard, that complement regulatory protein, that's going to make sure that complement doesn't get activated in absence of something foreign. Okay, so I told you I was going to bring this up again. So our weapons for fighting microbes, we have complement, and we know that complement is going to opsonize things, and then we get phagocytosis for them to be eliminated. So remember, we talked about disease and how one of the big things that microbes have to do is to resist the immune system. And so these are our weapons. And so Streptococcus pyogenes, remember, it can avoid phagocytosis. The ways it can avoid phagocytosis, one thing is it has a C5a peptidase. And so it can actually cleave and inactivate one of those complement proteins, and it happens to be the protein that stimulates inflammation. M proteins and capsules, remember how capsules, we said, make things harder for phagocytosis? That's because they attach the complement regulatory protein, it binds to them, so complement gets inactivated when there is a capsule. Streptococcus pneumoniae also avoids phagocytosis. It does it in another way. It actually can inactivate C3b. And so these are some of the ways that the bacteria get around our immune system. So they can defend against complement, and they can defend against phagocytosis. We're going to wrap it up for the week here. I hope you have a great day, and we will come back next time and talk about phagocytosis, inflammation, I think we have fever left, and apoptosis. Have a good weekend.